Okay, so this is Practical 1 Study Guide Review Part 4. This is our last part of our Practical 1 to go over. This is going to cover cell anatomy and cell division. So again, I'm sharing my screen here, and what you're seeing is the study guide. So in this screen right here, this is showing all of the structures that you have to know within a cell. This is straight memorization, so I'm not going to go over everything here, but I am going to point out a few things. So let me get my little pen again. So over here, I'm going to circle in red where it says cytoplasm and cytosol. Okay, so cytoplasm is the jelly substance that is between all these different cell parts. And the liquid within the cytoplasm is called the cytosol. So if I point in here to right here in the middle of nowhere, where there's no organelles or no cell parts in there, that is going to be cytoplasm. But if I point to that same arrow or area and ask you what the liquid is, then you would tell me it's cytosol. Okay, so if you think of it like a jelly that you would have in your refrigerator to put on your biscuits, when that jelly sits in your refrigerator, it not only has the jelly portion of it, but a lot of times a liquid will start appearing at the very top. So that thick jelly portion is synonymous with the cytoplasm, and that liquidy portion on top of that jelly is synonymous with the cytosol. Okay, let me erase my marks here. The other thing to point out here is around the nucleus. So the nucleus has some different parts to it. So you'll see here, it has, here it is, the nucleus, whoops. The nucleus is the whole thing. So it's this whole entire purple ball that I'm circling right here. That is the nucleus. But within the nucleus, you had this outside skin of the nucleus. That outside skin is called the nuclear envelope. And then this ball inside the nucleus is called the nucleolus. Okay, so again, if I'm asking this on a practical, if I put a number, if I put a number here and I say, identify number 1, the entire structure would be a nucleus. If I put trying to get my eraser again. Or if I put that to identify this specific thing right here that I'm putting the red mark on, this specific organelle or cell part piece would be the nucleolus. Or if I put number 2, identify this covering or layer, that covering or layer would be the nuclear envelope. For the lab, you have to be able to identify these parts, but you do not have to identify the functions of any of these parts. You're going to be asked the function of these parts in your first lecture exam. Another thing to point out here is at the very top, these little finger extensions off the top, those are called your microvilli. So they're parts of this cell membrane that are going to come and they're going to stick up to increase surface area of the cell. So the cell, for instance, would have more surface area to absorb nutrients, for instance, when it's in your digestive tract. So those little pieces of the cell membrane that are sticking up or protruding out of the cell at the very top of this model are called your microvilli. Now you have 2 other cell extensions. So we just covered the microvilli. So in this picture, you're seeing the microvilli, and you're seeing cilia, and you're seeing flagella. So the cilia here are going to be these longer hair type extensions. And when you're thinking of cilia, I know it's kind of gross, but if you'll look up your nose, those hairs sticking down would be cilia. And then the only time in the human body that you have flagella, which is a long whip-like tail, is in the male when they're producing the sperm. So their sperm cells will have a flagella, which is a whipping tail that move those sperm through the male reproductive tract, and once it gets into the female, we'll move that sperm up the female reproductive tract. So make sure you can identify each of these cell extensions, the microvilli, the cilia, and the flagellum. Okay. Now, the next thing we're going to talk about is cell division. So every time you go to bed at night, you're going to wallow around in your blankets, and your skin is going to be abraded or rubbed on by those sheets and blankets and by your clothing. And when it does so, it sloughs off dead skin cells. So you need a way of producing new skin cells so that you don't break through your skin. And this is happening all throughout your body through your entire life, the replacement of old cells with new cells. And the way the body does this is through mitosis. Mitosis is a type of cell division for body cells, not sex cells, but body cells. So we're seeing here a depiction of mitosis. Let me get my pen here because I'm going to be pointing out a few things. Okay, so most of the time, your body cells are going to be in this G0 phase, okay? In G0 phase, a lot of times the cell is just undergoing normal cell operations and it's not growing. Then when the body triggers these cells to start getting ready to divide, those cells need to grow, they need to duplicate their information, and then they'll grow again. So once the body has told a normal cell, and you'll see, I'm just going to put a number 1 here, that normal cell number 1 is now going to enter into getting prepared for cell division. The first area it enters in, or the first phase it enters in, is called interphase. And interphase is all of this part of the circle right here. So right there, red line all the way around, all of that is interphase. And within interphase, you have 3 subphases or 3 subdivisions of interphase. The first one here is G1. G1 is where your cellular contents are going to be duplicated, because if we're going to split that one cell into 2 cells, we need to duplicate all the contents of that first cell so that when it splits, both cells have the correct cellular components. After the cellular components have been duplicated, then we enter into S phase. And in S phase, the DNA is copied, or DNA gets synthesized so that you have 2 copies of DNA. Then from there, you're going to enter into the G2 phase. In this phase, the cell is going to do its last growth phase, and your cell is going to double check the chromosomes to make sure there is not a synthesis error. If there is a synthesis error, then that cell will be marked for death so that the body can get rid of an abnormal cell. So let's look at this again. I'm going to put down some numbers here and ask you what phase this is. Okay, so if I put a number 1 right here and ask you what phase it is, you would tell me interphase. If I put a number 2 here and ask you what phase, you're going to tell me G1. What phase is in number 3? S phase. What phase is number 4? G2 phase. Okay, so now we're going to add some more information here. We've covered all of interphase. So now interphase gets the body prepared to go into mitosis, which is where you actually have the one cell dividing into 2. So all of this right here, this phase would be mitosis. So you're seeing that mitotic phase. You can either put mitotic or mitosis. And in this mitotic phase, you have the actual mitosis, which is dividing into 2 cells. And then you have cytokinesis. Cytokinesis is where the cytoplasm gets divided into 2. Okay. Guaranteed, you'll probably have at least 1 question on your practical, if not 2, from this, make sure you can label all of these different parts here. Now we're going to review mitosis in a little bit more detail. So mitosis has several different phases. I'm going to go further down in our study guide, then I'll come back to this picture. Okay, this is what I wanted to do right here. Okay, you can remember your phases by a mnemonic called IPMAT. So there's I, and then there's PMAT. Oop. Okay, so this one over here by itself, that is interphase. Remember, interphase is going to be where the cell is growing and duplicating DNA, getting ready to divide, but the cell is actually not dividing yet. PMAT are the phases of mitosis. So only PMAT. Interphase is not actually a phase of mitosis. Interphase, the I, comes before mitosis. Okay, so your PMAT is mitosis. The P stands for prophase. The M stands for metaphase. The A stands for anaphase, and the T stands for telophase or telophase. Okay, so we've covered interphase pretty good on the previous screen where we talked about G1, S, and G2. Now we're going to go through these 4 phases of mitosis: prophase, metaphase, anaphase, and telophase. So now I'm going to erase all this and we're going to go back up to that previous screen that we were on, about 2 pages up from this. Okay. So when you're getting into prophase, the thing that'll help you identify prophase is everything is pretty much still in the nucleus area. So you see right here, this is the nucleus area, but what you're looking for is you can see these little yarn strings. Those yarn strings are your chromosomes that have condensed, okay? So if you can see little yarn pieces in the nucleus area, you're in prophase. I will never ask you early prophase versus late prophase, okay? So here, this is our nucleus area, and you can see these dark yarn type pieces in here. If you can see those tight yarn pieces, then you know you're in prophase. And the same thing here. There's my nucleus area, and you can actually see little yarn pieces in here. That would make it prophase. In metaphase, these chromosomes, you can see they meet in the middle. Okay, they meet at the midline. So metaphase meets in the middle. So all your chromosomes are meeting right here in the middle. So that clues you in that this is metaphase. Let me erase all my marks and we'll go on to the next one. Okay, so that's prophase and metaphase. Now we're going to do anaphase. So anaphase, you have the chromosome strings or the sister chromatids being pulled apart, and you have some chromatids. I'll get my pen again. You have some chromatids that are being pulled right here to this part of the cell. And then you have these chromatids or sister chromatids being pulled to this end of the cell, but they're pointing towards each other. So when you're looking at it, it looks like a bunch of fingers pointing to each other. So it kind of looks like this right here. Okay, where you see those sister chromatids being pulled to opposite ends, and the 2 sets of sister chromatids are facing each other, but you can see a little bit of distance between them. This next one is telophase. In telophase, you have the sister chromatids all the way over on this side, and you have the other sister chromatids all the way over on this side, and you're usually seeing a little bit of an indentation into the cell. Okay. Sometimes you will, sometimes you won't, but you can definitely see that those 2 cells are starting to form here with sister chromatids in this half, and then another set of sister chromatids in this half. Cytokinesis. Cytokinesis is where that cytoplasm has developed a cleavage furrow that has gone all the way through the cell. So this cell is now completely separate from this cell, but they are still in close proximity because they just got through cutting into two different cells. So that would be called cytokinesis. Okay. Now, all of these pictures right after this, they are the same pictures we just went over, but they are blown up. So instead of going all the way over to these pictures again, I'm going to share another document with you. Okay, so this document I'll put in your practical 1 shell or module. So this is the model that we use in our laboratory. So if I gave you only a picture of this one in number 1 and ask you to identify this phase, number 1 would be interphase, because as you can see inside this nucleus right here, there is nothing going on in there. Okay, he's just undergoing his normal everyday life with nothing much happening. So that would be interphase. Number 2 and number 3. In here, this is the nucleus area. This is the nucleus area here, and you can now see the little yarn pieces within that nuclear area. So these would both be prophase. You're seeing an early versus a late, but again, I would never ask you early versus late. I would just tell you that this is prophase. So again, the first picture or the first part of this would be your interphase. So this one here is interphase. Both this one and this one are prophase, because again, in the nuclear area, you're seeing the yarn pieces. Now this one is going to be metaphase, because you can see here they're meeting in the middle. This one also is going to be metaphase. It's just a later form of metaphase, and they're still meeting in the middle. This one here is going to be anaphase. You're seeing your 2 sets of chromatids, and you can see these chromatids are pointing at each other. So these are pointing down and these are pointing up. Okay, so if you can see the 2 sets of chromosomes or chromatids and they're pointing each other still, then that is going to be anaphase. Then this one is telophase. At telophase, you're seeing the group of chromatids converge to far sides of either part of the cell, and you're starting to see that cleavage furrow or that pushing in of the cytoplasm. So that's telophase. And this here would be telophase with cytokinesis. So remember, cytokinesis is where we have that cytoplasm that had the cleavage furrow that has completely split the cytoplasm into 2 different cells, cell 1 and cell 2 here. Okay. Now let me erase all these. And we're going to review a few more just so this is perfectly clear. Okay. So going down to the next screen. Oop, I think I went too far. Hang on. Here we go. So now let's look at what these would look like under the microscope. So you can see this interphase right here. This interphase, there's nothing going on in it. I mean, if you can't see anything going on in the nucleus area, then it's in interphase. This next one, I would not ask you this interphase beginning right here, because it's too hard to identify. So I would never ask you this one right here. I would go on to this one and this one and this one. These are all great examples of prophase. Remember, you're in prophase, you're looking for the little yarn strings inside the nuclear area. So all of those would be prophase. Do not worry about telling me early, mid, or late. They're just prophase. Then down here, picture number 6, that's going to be metaphase, because remember, metaphase, they meet in the middle. Then number 8 is going to be anaphase, because remember, you're seeing those little spiky sister chromatids pointing at each other. So these are pointing down and these are pointing up. So that would be anaphase. Then in early telophase, this set of sister chromatids has moved to one pole of the cell, and these sister chromatids have moved to the opposite pole of the cell. Then here you can see the cleavage furrow has now completely segregated or separated the cytoplasm. So that would be telophase with cytokinesis. Okay, moving down from this one. Okay, so I'm going to get my pen out here. I'm going to mark some of these and I'm going to see if you can identify them. So I'll ask you what phase it is, and then I'll pause and let you answer. What phase is number 1? That would be interphase. This one right here, what phase is number 1? That would be interphase. This one here, what phase is this one? That would be prophase, because you're seeing the yarn pieces in the nucleus. Number 3. Number 3 would be prophase as well, because again, you're seeing the yarn pieces. This would be another. Number 4 would be another prophase. Number 5 would be another prophase. Okay, so that gives you some examples. Now let's go on. Let's see. What would number 6 right here? What would number 6 be? Number 6 would be metaphase, because remember, they're meeting in the middle. What is number 7 here? Number 7 is anaphase. What's number 8? Number 8 is anaphase. What is 9? Telophase or telophase. And number 10? Telophase. And then this one right here, telophase with cytokinesis. Okay. Now moving on to some more examples. Okay, and this one again, I'm going to make a mark or a number on a cell, and I want you to tell me what phase it is. So number 1, can you see that? Number 1 would be interphase. Let's do number 2. 2 would be anaphase. What's number 3? 3 would be anaphase. What's number 4? This would be telophase. And in this example, we're not seeing metaphase in there. Okay, so that would be all of the examples. And again, I will put these mitosis images in the practical 1 module, as well as this video recording. Good luck on your practical.